Good evening, Jyoti. Can you able to hear me?
Yes, ma'am.
Okay. So let us wait for 2 minutes, then I will start the session. Okay. So we changed the timings from 6 to 7. As you said, the college timings will be till 5 o'clock. Why others are not attending? Any other reasons? You can message me in the chat box or you can tell me. Okay, let us begin. Sit in a comfortable position and please switch on your video while session is going on. Fine. So in the last class, we discussed regarding microsporogenesis, right? How the microspores are formed, means how the pollen grains are formed. So now our plant is ready with the microsporogenesis. The microspores are ready, right? Now female gametophyte or female reproductive part should be ready. Just a minute. We learned regarding male reproductive part, as well as we learned regarding how the microspore or a pollen grains are formed. That is by a microsporogenesis, right? Then we learned structure of anther in it, structure of anther, then microsporogenesis we learned. Okay. Tetrad, structure of microspore. Then we learned regarding function of pollen kit. Today we are going to learn yeah. So Jyoti, whether microgametogenesis got over in the last session? Whether microgametogenesis or development of male gametophyte got over in the last session?
Yes,
Okay. So today we are learning regarding the female reproductive part, which is also called as gynoecium. So we know that the female reproductive part in the flower is nothing but it is called as pistil. So it is composed of, the whole thing is called as pistil. A single unit of it is called as carpel. Pistil is the female reproductive part. Adralli single unit ge we will call it as carpel. Together pistil, single unit is called as carpel. So the pistil will be consisting of mainly three parts, Jyoti. The first one is stigma. This is a stigma, which is a receptacle, right? The next one is style. The next one is ovary, right? Stigma, style, and ovary. So inside the ovary, so ovary is there, no, this part. After the fertilization ಆದ್ಮೇಲೆ ovary ಏನಾಗುತ್ತೆ, it will be converted into fruit, right? So inside the ovary, there will be a soft cushion-like tissue. Inside the ovary, a cushion-like, a very soft cushion-like tissue, which is called as placenta. Cushion-like tissue, which is called as placenta. Placenta will be holding certain small structures which are called as ovules. certain small structures which are called as ovules, or it can also be called as megasporangia. It is also called as megasporangia. Ovules ಇನ್ನೊಂದು ಹೆಸರೇನು megasporangianu ಅಂತ ಕರಿತೀವಿ, right? So this megasporangia, it will be having its own structure. So ovules number may change. Some of the ovary will be having single ovule. For example, mango, wheat, paddy, right? ಅದರಲ್ಲಿ ಏನಿರುತ್ತೆ ಒಂದೇ ovule ಇರುತ್ತೆ. But when in case of watermelon, in case of cucumber, then in case of muskmelon, karbooj ಅಂತ ಏನ್ ಹೇಳ್ತೀವಿ. So ಇದರಲ್ಲಿ ovules will be more. So let us learn about the ovule. Now we came till ovule. Now we have to learn what is ovule, what is the structure of ovule. Ovule ಹೇಗಿರುತ್ತೆ? A ridge or a stalk-like outgrowth is formed from the placenta of the ovary on which body of the ovules are present. Each ovule is attached to the placenta by means of thin stalk called as funicle. So here you can see, if this is a placenta, the ovule, this structure is a ovule. The whole thing what you are seeing, no, the diagram is a ovule, right? So the stalk of a ovule is called as funiculus or funicle. We can call the stalk of ovule as funicle. Then the point of attachment of funicle to the placenta. So this ovule will be attached by a stalk in the placenta ge hatkondirutte. So this stalk is called as funicle. The point of attachment of the funicle of the ovule to the placenta, a point ge we will call it as hilum. That point of attachment of the funicle to the placenta or with the ovule is called as hilum, right? The main region of the ovule is composed of mass of parenchymatous cells called nucellus. So you can see here nucellus, yeah. So the ovule is consisting of mass of parenchymatous cells. So this region is called as nucellus, which provides nutrition to the developing embryo. Nucellus is the main part of the ovule. The nucellus again is covered by, as you can observe, the nucellus. This part is nucellus. Inner part is there, no? Inner part is made up of many cells, which is called as parenchymatous cells. So this region is called as nucellus. Nucellus is not, the cells are absent at one end. Nucellus layer alli, nucellus is made up of mass of cells. So adralli, it is free at one end. That free end is called as micropyle. Nucellus yava kade, yava dikkinalli means in which direction it is free, no? In which direction the cells are absent in the nucellus, that opening is called as micropyle. Okay. So this nucellus is there, no? It provides nutrition to the embryo, developing embryo. And the nucellus is covered by, several layers. These layers of ovule are referred as integument. These layers of ovules are called integuments, outer integument and inner integument. Then a place from where funicle and integuments arise is called chalaza. So here you can see one end. The integument arise. Illinda enagate, the layers and the funicle will arise. Illinda, e end inda, the funicle and the layers of ovules will arise. Arise andre allinda origin agate anta. So that end is called as chalaza. That end is called as chalaza. Or simply if you want to remember andre, micropyle elliratte, adar opposite end we will be having chalaza usually. Next, integument is absent just opposite to the chalaza. In one opposite to the chalaza, integument is absent. That opening is called as micropyle. That pore, tooth irutte. Okay, so that pore is called as micropyle. Then we have the placenta. They gave the definition of placenta here. Placenta means what? It is the cushion-like structure to which ovules are attached. So if you consider this as ovary, so this is placenta, and through the funicle, the ovules will be attached like this. I hope you got it, Jyoti. Did you get it?
Yes, sir.
Okay. Next. Please note down simultaneously. Okay, please note down. Next. Then we have types of ovules. types of ovules are there? This is based on integuments. Andre ovules sutta ಎಷ್ಟು layers ಇರುತ್ತೆ, ಎಷ್ಟು integuments ಇರುತ್ತೆ, ಅದರ ಆಧಾರದ ಮೇಲೆ we have, I think, 3 types of ovules. The first one is unitegmic, second one is bitegmic, third one is ategmic. Uni refers to single. So if the ovule is covered by only single integument, only one integument, then it is called uni. If a ovule is covered by 2 integuments, 2, so it is called as bitegmic. Bi means 2, tegmic means integuments. Okay, then so usually e type ovule ನಮಗೆ ಎಲ್ಲಿ ಸಿಗುತ್ತೆ, ಕ್ಯಾಪ್ಸುಲಾ ಅನ್ನೋ ಎಕ್ಸಾಂಪಲ್ ಅಲ್ಲಿ ಮತ್ತೆ ಮೋನೋಕಾಟ್ಸ್ ಏಕದಳ plants ಅಲ್ಲಿ we can observe bitegmic ovule. Next, the ovule in which integuments is called, integuments are absent, ovule sutta layers illde hodre, integuments are totally absent idre, then it is called as ategmic ovule. Then it is called as ategmic ovule. So based on integuments, the ovule is divided into 3 types: unitegmic, bitegmic, ategmic. Means absent of integument, right? Then on the basis of nucellus, the mass of tissue, again the ovule is divided into 2 types, mainly 2 types. The first one is tenunucellate. The second one is crassinucellate. Tenunucellate means what? The nucellate is either less developed or present in the form of single layer. So if this is a embryo sac, if this is a egg cell, the nucellus is single layer idre, the mass of cell is single layer, then it is called as tenunucellate. Crassinucellate andre the nucellus is massive type. It is in the form of a layers of cell. It is in the form of a layers of cell. So that is called as crassinucellate, right? So after fertilization, what happens? The nucellus tissue will degenerate. And integument, the layer becomes active to form nucellus-like tissue, which is called as endothelium or integumentary tapetum in the ovary. This is regarding the classification based on nucellus. Next, we have on the basis of type, very important, on the basis of relationship of micropyle, chalaza, hilum with the body of the ovule and orientation of it. So micropyle, chalaza ಮತ್ತೆ hilum ಹೇಗೆ ಅರೇಂಜ್ ಆಗಿರ್ತವೆ, ಯಾವ ಅಲೈನ್ಮೆಂಟ್ ಅಲ್ಲಿ ಇರ್ತವೆ, ಅದರ ಆಧಾರದ ಮೇಲೆ we have around A, B, C, D, E, F, 6 types of ovules. The first one is orthotropous. The first one ovule is orthotropous. Orthotropous andre enu? As you can see, the micropyle ಮತ್ತೆ chalazial end, they will lie in the same plane. They will lie in the same line. Micropyle, chalazial end and the embryo sac, that is female gametophyte. So all 3 lie in the same plane. Such a type of ovule is called as orthotropous ovule. Next, when you come to the sarcinotropous ovule, sarcinotropous means what? Here, the funicle itself will be surrounded. The funicle itself, the micropylar end will be inner. The funicle itself will be surrounded in the form of a circle around the ovule, which is called as sarcinotropous. The next one is anatropous, most common type of ovule. So here micropylar end will be below at the bottom, chalazial end will be at the upper side, and micropyle and funicle lie next to each other. ಇಲ್ಲಿ ಏನಾಗಿರುತ್ತೆ, ಮೈಕ್ರೋಪೈಲು ಮತ್ತೆ ಫ್ಯುನಿಕಲು, they will be adjacent, ಪಕ್ಕದಲ್ಲೇ ಇರ್ತವೆ. Next, hemianatropous. Hemianatropous ಅಂದ್ರೆ ಏನು? This is a funicle. The ovule lies right angle. See here, the ovule lies, this is a right angle, 90 degree, right? So the ovule lies right angle to the funicle. This is a funicle. This is a funicle, right? The ovule lies with it. So this is a right angle, right? So the ovule lies right angle to the funicle. This is called as hemianatropous. Amphitropous. Amphitropous means what? Here the embryo sac as well as the body of the ovule, embryo sac as well as the body of the ovule will be curved. So that is called as amphitropous. So here what happens? The micropyle, hilum and the funicle will be lying adjacent to each other. Right. Then after that, campylotropous. Here only body of the ovule will be curved, but not the gametophyte. That is, the embryo sac will not be curved. Here see, the embryo sac is also curved, right? So here embryo sac will not be curved. It is same only, like this only. So such ovules are called as campylotropous ovule. Orthotropous, sarcinotropous, anatropous, hemianatropous, amphitropous, and campylotropous ovules. Okay, Jyoti, any doubts in this? Yes, Jyoti, any doubts in this? Are you here, Jyoti? Okay. Okay, fine. Let me proceed further. Now we have megasporogenesis. Megasporogenesis. So let us watch one video. So through that, you will understand the, just a minute, yeah. See, this is a flower and this is a ovary, what they are showing. Find the ovary, then flower, stamens, you can see anthers, and this is a gynoecium. Each individual, each part is called as carpel. Together it is called as pistil. Right, this is a bottom part, which is a ovary. Ovary will be having a stalk, which is called as style. At the tip, we will be having a receptacle, a sensory part, which is a stigma. Stigma, style, and ovary. Right. Jyoti, whether the video is playing?
Yes, sir.
Okay. So when you go inside the style, when we proceed, travel inside the slide, style, so we will get a bulge one, which is called as ovary. Let us see what is there inside the ovary. See how beautifully the ovules, the megasporangia are arranged in the ovary. So these are nothing but a megasporangium or plural megasporangia. These are the integuments, the layers. Okay. So these are nothing but the ovules we can call. So in the ovule, the opening, the opened pore is called micropylar region, and the stalk green color what you are seeing, no, it is a funicle. This whole thing is a ovule, right? This is a one ovule what we are seeing. So inside the ovule, there will be one megaspore or sporocyte. There will be one megaspore. That megaspore will undergo meiotic division. And you can see here, after meiosis, we got 4 spores. After meiosis, we got 4 spores. So megaspore mother cell undergo meiotic division and formed 4 megaspore cells, out of which 3 will degenerate. You can see 3 got degenerated. The one is remaining, which is a functional megaspore. Idenidu functional megaspore. Again, just a minute. This megaspore will undergo nuclear division. This megaspore will undergo mitotic division, where only nucleus will be divided. Fine. So here you can see the nuclear division happens. Just a minute. Okay. Just a minute. Okay. So this video we were watching. So here what happens? So here one megaspore will be there. That one megaspore, it got divided, underwent mitotic division and formed 4 megaspores. One megaspore mother cell undergo mitotic division. Undergo meiotic division and formed 4 megaspores. So megaspore mother cell enittu diploid ittu. So when it undergoes meiosis, we got 4 haploid single set megaspores ಸಿಕ್ತು. So 4ರಲ್ಲಿ 3 will degenerate and 1 will be functional. 4ರಲ್ಲಿ 1 functional ಆಗತ್ತೆ. After this, that one megaspore will be there, no? That one megaspore will undergo mitotic division. Megaspore mother cell meiosis ಆಯ್ತು. Now that one functional megaspore will undergo mitotic division. ಯಾವ ರೀತಿ mitotic division? In this mitotic division, there will be only division of nucleus. ಅಲ್ಲೇನಾಗತ್ತೆ? Only division of nucleus ಆಗತ್ತೆ. Cell will not divide. Means cytoplasm divide ಆಗಲ್ಲ. There is no cell wall formation. So what happens? One megaspore undergo mitosis, it forms 2 cells. Again 2 cells undergo mitosis, it forms 4 cells. Again 4 cells underwent mitosis and forms 8 cells. So totally 8 cells ಆಗೋವರೆಗೂ ಈ ರೀತಿ ನ್ಯೂಕ್ಲಿಯರ್ ಡಿವಿಷನ್ ಆಗತ್ತೆ. ಎಲ್ಲಿ? ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಅಲ್ಲಿ. Right, you can see. Yeah. So now we have 8 cells. Yes. Now we have 8 cells in the embryo sac, out of which 2 cells, 1 cell from the upper region, 1 cell from the lower region, they will come at the center. ಒಂದು ಸೆಲ್ ಕೆಳಗಿನಿಂದ ಇನ್ನೊಂದು ಸೆಲ್ ಮೇಲಿಂದ ಬಂದು ಸೆಂಟರ್ ಅಲ್ಲಿ ಲೊಕೇಟ್ ಆಗತ್ತೆ, ವಿಚ್ ಆರ್ ಕಾಲ್ಡ್ as ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈ. ಸೊ 2 ಸೆಲ್ ಸೆಂಟರ್ ಅಲ್ಲಿ ಬಂದಾಗ ಹೌ ಮೆನಿ ಸೆಲ್ಸ್ ಆರ್ ರಿಮೈನಿಂಗ್ ಅಟ್ ಬೋತ್ ದ ಎಂಡ್ಸ್ ಜ್ಯೋತಿ? 4ರಲ್ಲಿ 1 ಸೆಲ್ ಮೇಲಿಂದ ಬಂತು, ಇನ್ನೊಂದು ಸೆಲ್ ಕೆಳಗಿಂದ ಬಂತು. It formed the polar nuclei. Now tell me how many nucleus are present? Nuclei are present at both the ends, Jyoti?
3.
Very good. So three nucleus are there at the both ends. So lower side alli enagatte, 2 cells will form synergid, 1 cell will be forming egg cell. Upper side alli all 3 nucleus forms 3 cells which are called as antipodals. You can see here in the video now. See 8 cell aita, 2 cell naduve bantu. These are called polar nuclei. 3 cells enagide? 3 cells. So out of 3 cells e end alli 1 will be egg cell. Okay, the other 2 will be synergid cells. And at the other end 3 nucleus were there. Polar nuclei, they fuse to form central cell. Polar nuclei fuse ಆಗ್ತವೆ to form central cell. Other end alli 3 nucleus forms 3 cells which are called antipodals. So how many cells we have now? 3 antipodal cells, 1 egg cell, 2 synergid, 6, 1 central cell. 7 cells ಆಯ್ತು. How many nucleus we have now? 3 antipodal cells to 3 nucleus. ಆಮೇಲೆ synergid 2 nucleus, egg cell 1 nucleus. So 3 plus 3 6. Polar nuclei are 2. 6 plus 2 8. So totally our embryo sac or female gametophyte ಎಷ್ಟಿದೆ? ಅದರಲ್ಲಿ 7 celled 8 nucleated. Embryo sac is 7 celled 8 nucleated. ಹೇಗೆ? Just a minute. Okay. Okay. So after this, yes. So here we got the embryo sac. We got the embryo sac or a female gametophyte. So ಇವೆಲ್ಲ process ಆಗ್ತಾವಲ್ವಾ, megaspore mother cell ಇಂದ we got megaspore, functional megaspore. So that megaspore underwent mitotic division and formed one egg cell, two synergid, two polar nuclei and three antipodal cells. So cells anta bandaga, three antipodal cells, very important for CET and NEET. Okay, three antipodal cells, one egg cell ide, two synergid cells are there, right? Then two polar nuclei were at the center. They fused and formed central cell. Averadu fuse agi, they form central cell. So how one central cell is there? 3 plus 1, 4, 4 plus 1, 5, 5 plus 2, 7. So female gametophyte alli athava embryo sac alli, how many cells we have? 7 cells we have. Right. Now let us come to the nucleus. Synergids ge tannade adantha nucleus irutte, egg cell ge tannade adantha nucleus irutte. So 3 nucleus at this end. Right. Next here, 3 antipodal cells have 3 nucleus. 3 plus 3, 6. Then 2 polar nuclei are there. No doubt they fused, but they have 2 nucleus, right? So 2 nucleus. 3 plus 3, 6. 6 plus 2, 8. So 8 nucleated. So embryo sac enide? Embryo sac has 8 nucleus and 7 cells. I hope you got this point, Jyoti. Did you understand this? Okay, good. Fine. So it has been asked in NEET also. It has been asked in the this CET also. So this is regarding the megasporogenesis. Megasporogenesis. Now, can you read out this, Jyoti?
Yes, ma'am.
You can go to other place and you can read out. Otherwise, you can read out like that only. No problem.
The process of formation of megaspore from megaspore mother cell by meiotic division is known as megasporogenesis. During the development of ovule, in the beginning of this process, nucellus develops from the placenta in the form of a small rounded outgrowth-like structure. At this stage, all the cells of nucellus are undifferentiated and homologous and meristematic. This mass of cell is surrounded by single-celled thick layer of the epidermis.
Okay, Jyoti, 1 minute. 1 minute. Good evening, Mahalakshmi. Welcome to the session. You came at 6:30. Fine. Can you read out the concept, please? We discussed megasporogenesis. Can you read out the concept? Unmute yourself and read out the concept, please. Please don't go out of the session because ma'am asked to read. However you want to read, you can read it. You can start from a single hypodermal cell. Mahalakshmi, are you here? You can message me if you have any problem. The PPT is already shared on the screen. PPT is already shared on the screen. You can see the PPT now. Just check. See, if you don't have a network, you can't attend the session. You have the network, you have everything, but you don't have a mind to read. That's it. Okay. Jyoti, continue from a single hypodermal cell.
A single hypodermal cell of nucellus is differentiated and increased in size. It becomes different from the rest of the cells due to presence of distinct nucellus. It is called as
Archisporial cell.
archisporial cell. Archisporium divides
Periclinally.
periclinally to form an outer primary
Parietal cell.
and inner primary sporogenous cell. Activity of primary
Parietal cell.
parietal cell depends a type of plant.
Okay.
A plant belongs to
Gamopetally.
gamopetally. Then it forms
Tenunucellate.
tenunucellate type ovule. And if plant belongs to polypetylate, then it forms
Racenucellate type of ovule.
the primary sporogenous cell directly acts as a megaspore mother cell. It is diploid and divides mitotically to form 4 haploid megaspores. The 4 haploid megaspores generally arranged in linear tetrad. Generally, the lowermost or chalazal megaspore remains functional out of tetrad of megaspores, and the other 3 which lie toward the micropyle degenerate. This functional megaspore produce female gametophyte. In most angiosperms, chalazal megaspore remain functional.
Okay, thank you, Jyoti. So now we will discuss certain MCQs, which I will be dictating you from the book, one of the book. You have to note down it. Just a minute. Yes. Hmm. Okay. Yes. The first question here is, the tapetum is found in. The tapetum is found in. The first question is, the tapetum is found in. Write down, tapetum is found in. Is found in. Option A, anther. Option B, androecium. Option C, ovary. Option D, ovule. Tapetum is found in anther, androecium, ovary, and ovule. This is the first question. Did you write, Jyoti? Okay. So you have to send it or mail it. I will give you the number. You have to mail it or send it to that number. Okay. Shall I proceed to the next question? Shall I delete this?
Wait. Okay, ma'am.
Okay. Next. Megasporangium. Second question. Megasporangium Megasporangium, along with its protective integuments, along with its protective integuments, along with its protective integuments is called Megasporangium, along with its protective integuments is called option A, ovary, option A, ovary, option B, ovule, option B, ovule, option C, funicle, option C, funicle, option D, chalaza or chalaza. Did you complete writing the question, Jyoti?
Yes, ma'am.
Okay, I will write the options. Option A, ovary. Option B, ovule. Option C is funicle. Option D, chalaza. Okay, chalaza length. Write down. Once you completed, let me know.
Yes, ma'am.
Okay. Next one. Mature ovules, third one. Mature ovules are classified on the basis of matured ovules are classified on the basis of funiculus. This is a half part of a question. Funiculus. Next, if micropyle lie close to the funicle, lie close to the funicle, the ovule is termed as. Once you completed writing this, let me know.
Yes, ma'am. Completed.
Okay. Funicle, the ovule is termed as. The ovule is termed as. Option A, orthotropous. Option B, anatropous. Option C, hemitropous. Option D, campylotropous. Campylotropous.
Yes, ma'am. Completed.
Okay. Fourth question. Fourth question. non-essential floral organs in a flower. Non-essential floral organs in a flower. Are option A, sepals and petals. Option B, anther and ovary. Option C, stigma and filament. Option D, petals only. Option D, petals only. A, sepals and petals. B, anther and ovary. C, stigma and filament. D, petals only.
Yes, I complete.
Good. Fifth question. The stamens represent A, option A, microsporangia. Option B, male gametophyte. Option C, male gametes. Option D, microsporophylls. Microsporangia, male gametophyte, male gametes, microsporophylls. Fifth question. Okay. Now, so Jyoti, these are the 5 questions which are from NEET. Many other questions are there. Fine. So you have to complete it by today and send it before 9 p.m. today. So I will be messaging you the mail ID. Can you mail this, Jyoti? You can take the photograph and mail it. Can you mail it, Jyoti?
Yes, ma'am.
ನಿನಗೆ ಗೊತ್ತಲ್ಲ ಹೇಗೆ ಮೇಲ್ ಮಾಡೋದು ಅಂತ?
ಹಾ, ಗೊತ್ತು ಮ್ಯಾಮ್.
ಓಕೆ ಫೈನ್.
ಮ್ಯಾಮ್ ನಿಮ್ಮ ಮೇಲ್ ನನಗೆ ಹೇಗೆ ಗೊತ್ತಾಗುತ್ತೆ?
ನೋ ನನ್ನ ಮೇಲ್ ಅಲ್ಲ, ನಿನಗೆ ಮೇಲ್ ಕಳ್ಸೋದು ಹೇಗೆ ಗೊತ್ತಲ್ಲ ಅಂತ ಕೇಳಿದೆ. ನನ್ನ ಮೇಲ್ ನಾನು ಕೊಡ್ತೀನಿ ಈಗ ನಿಂಗೆ.
ಹಾ, ಓಕೆ ಮ್ಯಾಮ್.
ನಿನಗೆ ಮೇಲ್ ಕಳ್ಸೋದು ಹೇಗೆ ಗೊತ್ತಲ್ವಾ?
ಹಾ, ಗೊತ್ತು.
ಓಕೆ. Just a minute. Okay. Sankalp learning@gmail.com. So I messaged my ID, Jyoti. Just check it. Did you get?
Yes, sir.
Yeah. Please write it down. ಯಾಕಂದ್ರೆ ಆಮೇಲೆ ನಿನಗೆ ಇದು ಸಿಗಲ್ಲ. Once you open the chat box, write the spellings correctly so that you can send by 9 p.m. today. You have to send. Only 5 questions I have given, Jyoti. Okay. You have to write the question. You have to write the option. You have to tick the correct answer. Can you do it, Jyoti?
Hi, yes, ma'am.
Okay. please tell your friends to attend the session. Why? Whether they are not satisfied with the teaching. ಇಷ್ಟ ಆಗ್ತಿಲ್ವಾ ಅವರಿಗೆ ಸೆಷನ್? ಸೊ ಯಾಕೆ ಅಟೆಂಡ್ ಆಗ್ತಿಲ್ಲ ಅವರು?
ಗೊತ್ತಿಲ್ಲ ಮ್ಯಾಮ್, ಅಷ್ಟೊಂದು ಯಾರು ಪರಿಚಯ ಇಲ್ಲ ಮ್ಯಾಮ್.
ಓಕೆ 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 ಫೈನ್. Did you write the mail ID?
Yes, ma'am.
Okay, let us wind up the session here. Okay. Take care. Bye-bye.